Hello, everyone, and welcome to this week's edition of Journal Club. We will be discussing the research paper meta-analysis of sperm DNA fragmentation using the sperm chromatin structure assay. Here's a quick agenda of what we'll be talking about today. So we'll do the research paper review, and then we'll have a discussion. The audience is welcome to unmute your mics and put on your camera, and then we'll give you a little sneak peek on what's to come. So just to make this an easygoing experience for everyone, please update your name to reflect your first name as your first name and your last name as the city you are in. Also, feel free to submit your questions or just come off camera, raise the hand, whatever you want to do. And just know you'll get a recording of this when we're through. So let me turn it over now to Dr. Tony Anderson, who will be your host today.
Just come in here if you like. Hey, everybody. Thank you for being here today. I'm very excited to be sharing this with everybody. This is something we like to do with our students at the IVF Academy, and we've talked about some conventional versus ICSI. We did some in vitro maturation talks last time. This time we're going to talk about a celebrated scientist, and doing this bit in memory of Don Evenson, who passed away this past year. Don Evenson was a pioneer in the field of andrology. He had more than 200 peer-reviewed science publications that have been referenced over 14,000 times. And if that doesn't give you goosebumps, nothing will. I mean, he really worked hard, and what I really loved about Don, I had the privilege of knowing him and seeing him at meetings. I never did any personal work with him. I do have some colleagues that had done some personal work with him during their PhDs at EVMS, and just an amazing guy, and had a passion for what he did. And so I think this is probably one of his last papers, not one of his last. This was published in 2006. So, but it's a meta-analysis of this. And what I liked about this was it was basically a summary of a number of papers. We're doing some work at the IVF Academy with DNA fragmentation using the HALO assay. And I'll share a little bit of that data with you here in a little bit, but he really got the discussions going back in the in the 90s. But if you look at some of the early pioneers, Aitken did some of this work in the late 70s, early 80s, the very first papers on DNA fragmentation. And at the end of the day, where does DNA fragmentation come from? And so it's something, and I always say that sperm is the forgotten gamete. Sperm is the forgotten gamete. We like to focus on on the eggs and what's going on with those, but 50% of what we are working on our patients are, the contribution is the male. And if you were to break down infertility, we say that 40% of the infertility comes from male factor, 40% from female factor, and the other 20% comes from an idiopathic, if you're just to break it all down. And what does that mean? Where does that come from? And if you look at, this paper doesn't really address what percentage of the population has elevated DNA fragmentation. And so, what does one do if they want to know? You do a Google search. And so did a Google and it's not that it's totally completely precise, but if you did a Google search and says what percentage of the population has high DNA fragmentation in the sperm, it said that 80% of unknown infertility is caused by DNA fragmentation in the sperm. Now, I don't know if that's accurate or not. I will share some of our data with that, but let's just open it up to discussion. I know we have the Positive team and my IVF Academy team in San Antonio, and and I'm actually in Houston today, so I'm going to ask them if they could maybe open it up to discussion and see what their thoughts were on the paper.
there was a large data set, which was nice. I guess on the analysis wise, like they use, you know, the SCSA and the tunnel and the comet. And I guess it would have been nicer if it was all the same assay rather than different kinds. That's just my personal opinion on that.
Yeah, they were just comparing, you know, the tunnel assays and the different types of assays, and basically describing that that the sperm chromatin structure assay (SCSA) was a good cert test. And they, you know, I mean, basically Don had done a lot of his work, and other people had done, and you know, you could even go back to the Halo assay, right? And like, how accurate and how good is that test? And he's saying that it's a good test, comparing it to those. I do believe a lot of people believe that the tunnel assay is probably the gold standard, but I don't know if that's true or not. What was that? Can you repeat that?
It may have just been background noise.
Okay. Any other comments?
I found it it was a very interesting read, but I think one of the limitations that it has is that it tracked pregnancies, but it didn't track like long-term outcomes or like births or the health of the offspring. I didn't find that in there, so I mean.
Yeah, you're right. There was some comments talking about how infertility in general, there's increased risks of birth defects, and it doesn't really say specifically from ICSI, but just IVF in general compared to the natural population. And I believe it referenced a 2005 paper with that. And you know, I think some people have been concerned about that with ICSI, and we talked about this a couple weeks back in another journal club. Like, are we inducing problems with ICSI? Is it causing some imprinting errors? You know, I think when you're working with a male factor population, it's unique. You almost need to put them in a category of their own in the sense of apples and oranges. Historically, someone with male factor pre-ICSI days couldn't have a baby, whereas today they're having babies. People are able to have their own biological children with, you know, with very minimal sperm. But where does DNA fragmentation come from? It doesn't really talk about this here in the paper, but where does it come from? Anybody have any thoughts on that? Did you dig down into that?
Well, I did. I'm sorry. I did. I did look into it, and it says like with aging, like the DNA fragmentation like just increases in general. And there were like some other theories saying like the like smoking and like lifestyle, polyester even, like floating with polyester can like decrease or like I mean like damage more the DNA in the sperm, like just a lot of things that might be taking part of the DNA fragmentation being like higher.
Yeah, that's a good point. I mean, DNA fragmentation starts occurring from the very early stages. It could actually happen inside the testicle, right? And so before it's even ejaculated, the DNA fragmentation could be occurring. So lifestyle, like you said, smoking could cause that. You know, what do they do for work? Are they exposed to high temperature environments? I had a patient one time who was a baker, and he had exposed to really high temperatures, and he had, you know, extremely low sperm counts. This is pre-ICSI days. That was back in the day when I would have a guy produce 2 or 3 samples in a day just to get enough to do conventional insemination. And so, you know, if we would have had some of this technology back then, would we, you know, we may have even been able to identify more with those guys. As a matter of fact, it did talk about this a little bit in the paper, where it said that, you know, sperm concentrations and motilities, it's kind of across the board. What, you know, does it influence DNA fragmentation? But, you know, what I would like to say is if you had someone, like, one of the things we haven't mentioned here is that what really came out of this was that if it's greater than 30%, if DNA fragmentation is greater than 30%, or the sperm chromatin structure assay had a severe abnormality of greater than 30%, there was a significant drop in pregnancy rates with IUI and natural conception. What we did notice is that I did some work with this years ago when I lived in North Carolina, probably in the early 2000s. We discovered that if we did ICSI on these people that had high DNA fragmentation, that the pregnancy rates normalized. And so for me, back in those days, I said, well, let's just do ICSI on everybody rather than doing DNA fragmentation testing on everybody. Was that the right answer? I don't know. Going back. Kind of in light of that, I'll even let anybody in the audience kind of, if they want to unmute. I don't know if you're able to unmute, but give me some of your thoughts on that, if you would. Should we be doing ICSI just for DNA fragmentation? That rather, or should we be doing testing for DNA fragmentation in lieu of ICSI?
That's kind of how I took this paper. Oh, sorry, go ahead, whoever that is.
I personally think that we should, if we have the ability to combat DNA fragmentation, no matter if it's high or if it's low, why not? So, similar to us using Zymot or, Protex, using the best of the best, why would you risk it and take another approach and do a testing? And are we prolonging pregnancy in that way? How long does it take to get the results back? How much does it cost? For all of the different tests that we need to do on the sperm, can we just do ICSI and get it over with and get a baby here faster? Versus trying to find the sole reason as to why we're getting aneuploid embryos or not enough embryos in general. I don't know. It's just my opinion. But
oh, great. Yeah, thank you for that. That was kind of my answer to the question back then when we looked. Who's that? Say it again.
Hey, it's Ellie here. I was thinking about this DNA testing, and because it's a patient, how patients sometimes have insurance benefits and they have to do IUI before they go to IVF treatment. I think this testing will be beneficial if they get diagnosed with DNA fragmentation to move to IVF right away. That's where I would see it more beneficial.
It's a great point. Yeah. Especially if you're doing IUI and insurance is going to cover the IUI, where the data, Don clearly shows here that if you have high DNA fragmentation, IUI is the very, very low pregnancies. Great. Yeah, and I would say that if I were to, I do a lot of ICSI in my lab. We do 100% ICSI in our lab at Positive. And in all my labs, and this goes back to some of the work we did back in 2004, so over 20 years ago that we did some of this work. It was we sent samples to Don in his lab in South Dakota there, and had the analysis done. And if anybody had high DNA frag, we just did ICSI on them. That was back in the days when we did a lot more conventional. And so that's how I kind of migrated into doing 100% ICSI, because that was my way of circumventing, the not missing the high DNA frag person. And what we would actually send patients, someone who had miscarriage after miscarriage, multiple miscarriages. I think it even mentions in here more than 2 miscarriages, you'll find that they have higher DNA frag in their sperm. And so we would send that out and we would do ICSI on their next cycle. But, this has kind of migrated over the years. And I think Brittany mentioned that, Zymot, Novasort, there's another one, CA0 from Bonray Bio, Hamilton Thorne or Next Spring sells that. And I do believe IVF Store now has a product out with a note on that called Sperm. What's it called? Swim Count Harvester. So now, it used to be Zymot was the only device you could use for this. And actually, Danielle, who is here, works with the Academy, lead instructor there, has actually done some really great work, and her master's was on that. Can you forward to the next slide, I think? Yeah, this is Danielle's work where she basically took, and then Zoe Anderson, my daughter, I'll give her a shout out for that. We're still in the process of writing this paper, but we've taken now almost 50 samples of the raw ejaculate, separating it out through 3 different sperm preps using a gradient prep, swim up, and the Zymot, because Zymot was really the only device at the time. And we showed that, between the gradient and the raw sample, there's really no improvement in sperm quality as far as DNA fragmentation goes. I had some colleagues when we were presenting this data, I had some colleagues call me up like, why don't I just do a swim up? It's cheap. So we thought, well, let's take a look and see what happens. And there is a significant improvement. It actually goes down to about 10% DNA fragmentation. So almost a 50% improvement there. Zymot had actually less than 2% DNA fragmentation, and the data still holds true. We actually are doing some more work comparing this data to the NovoSort, and it's the same outcome. The NovoSort has a chamber that goes down into the specimen cup, and the sperm swim into it versus swimming up like the Zymot or the sperm harvester is, and so that sperm actually is still showing virtually 0 DNA fragmentation at different time points. I mean, I'm gonna say 0, like I would say less than 5%. Some is less than Zymot was around 2%. NovoSort's probably around 3 to 4%, but still significant improvement of sperm. So there's new ways of going about this so that we know we're giving it the best sperm in the lab. But kind of getting back to the paper, we know that if it's greater than 30%, maybe we should be doing some of this testing, especially if we're going to be doing IUIs. Any other comments from the students at the IVF Academy there? About the audience, anybody, what are their thoughts? I think you can, I do know that a couple other people were able to unmute. If you can unmute, you'll have the ability to unmute and share some of your own experiences possibly.
I actually wanted to say something. I agree with Brittany that it would be kind of costly and more time. I guess it would take more time, but I did look up what typical semen analysis would cost, and it would be around $50 to $150. But the SCSA test is around $300 to $500, but it prevents unnecessary IUIs or IVF attempts. And in the long run, it would potentially save money if they find DNA fragmentation.
It's a great point. Thank you for that. Yeah, I'm just kind of sitting here thinking as you're talking, you think about 40% male factor, 40% female factor, but let's just say you have someone who is, they're being diagnosed by the doctor, and they would go in and they would have their AMH done, their HSG done, and their semen analysis. They just can't find anything wrong. So maybe once you can't, that idiopathic group, you don't have to do it on everybody, but maybe do it on that 20% of your population that is, it's just not getting pregnant and you can't figure it out. You might do that. Carrier screening has been something that was only done on a few people. Now it's pretty much done across the board. to make sure we're not transferring any genetic abnormal embryos. One
So
thing that I didn't really talk about in here was that historically, what we would do in the past, if someone had high DNA fragmentation or repetitive failures with IVF or repetitive spontaneous abortions, when they had high DNA fragmentation or any of those other things I just mentioned, we would do testicular biopsies. And I would imagine that there's some people in the audience that have done that as well. What we found is that if you are doing this, what you're getting is that if the sperm in the ejaculate has high DNA fragmentation, the further you go up into the testicle, say the epididymis or the testicular biopsy, you would have lower levels of DNA fragmentation, but you're also swapping that with higher levels of aneuploidy or disomy in the sperm. So it's some give and take in the sense of doing that. I just had a talk with one of our docs the other day, someone who has had several failed cycles in another center. And historically, we would say, let's just do a testicular case. But I would like to try doing a cycle with just using one of these sorting devices and see what happens. Any thoughts on that?
Yeah, I actually have a kind of a question about your comment on you do 100% ICSI in your centers, right? All your centers. What is your sperm processing? What is your go-to, processing method?
Well, I would, kind of over the years, I was just like everybody else. Most people have done, once we discovered density gradient in the early 90s, we just used that for the next 30 years. It was around 2012, I started doing the Zymot prep, and actually it was easy to use, easy to teach, very consistent in the samples, 100% modal. So I started doing that around 2012. Been doing that ever since. Today, I'm actually doing some work with the NovoSort in the lab, but for the last probably 10 plus years, I've been doing the Zymot in my lab, and only for IVF cycles. I haven't really used it for IUIs. I think it could be, but I just haven't. I've used that. That's my go-to sperm prep.
Yeah, and we're
We're
offering it.
Slide, yeah. Go ahead. I'm sorry.
We're offering it to the patients, but they need to sign off that they're willing to use it. So we actually find that probably, I don't know, maybe 30% of the patients that are doing ICSI are actually approving the use of the Zymot. And I think with this data, that slide that you have there with the fragmentation, it's kind of hard to argue against it, right?
That's exactly the way I felt about it. I've also we've seen that with the other sorting devices that are out now too, very similar outcomes. So thank you for your comments. I really appreciate that. Could you go to the next slide, Lygia? One of the things that was a surprise discovery when we started using Zymot was, and we're still trying to figure this out. We're working on some research in the lab, in our training lab, looking at euploidy post Zymot prep, and also looking at maybe even like the swim up. Like I have some data on swim up preps from another lab I worked at. But surprisingly, and I think Palermo at Cornell, they published a paper not long ago showing similar results where they actually had higher rates of euploidy in the Zymot versus the density gradient preps. Now, historically, most of your aneuploidy is said to come from your oocyte and its first meiotic reduction of chromosomes, but maybe there's more disomy in the sperm than we know, or maybe just the quality of sperm, we're getting fewer mitotic errors. We don't really know, and I've actually had several conversations with folks. I think Jacques Cohen had mentioned, maybe it's the centromere that is contributed by the centrosome that's contributed by the sperm. But if I could, my whole goal with a lot of this journal club is that it encourages other people to possibly dig into some of these ideas. And this might be some of this DNA fragmentation might actually be old literature stuff, but I think as we get better and better with technology, it might be worth kind of redoing some of this research. So we started seeing more euploid embryos, and that's really why I stuck with Zymot all this time. This is actually some old data. I haven't looked at the data in a couple years since this, but once you get them pregnant, you can even see here. We actually, even though it wasn't significant, we had actually we actually saw no difference in embryo development rates either between the two different preps. So, I mean, you're not going to get better embryos, but you will get more embryos, but you will get better embryos. And so what was really interesting is that this is for the greater than 35, and that is the group that is really mostly needs it. And if you get to less than 35, there really wasn't a significant difference. And it makes sense because patients less than 35 should be making euploid embryos. But patients greater than 35, if you go from having 1 normal to 2 normal, that's the difference between, or 0 normal to 1 normal. That's the difference between having a baby and not having a baby. So, and it's really not about kind of, trying to find ways, about really promoting any kind of Zymot, but I think DNA fragmentation plays a bigger role in our infertility care than we really realize. And I think Dawn was one of the first people to do that and share that with us. And so, is anybody here doing DNA fragmentation in their lab regularly?
Hi, I don't know if you can hear me.
I can hear you.
Okay, perfect. I'm Emma from Ovation, and one of our physicians actually really believes in, DNA fragmentation, and she uses Zymot quite frequently with those with high DNA fragmentation, and we've seen drastic changes in pregnancy rates for IUI and both IVF with using Zymot.
Oh, that's great to hear that. Thank you for your feedback on that. I've only had my own personal experiences here, so I really appreciate it. Any kind of feedback like that. Maybe we should collaborate some of our data together, and I think that the more you can get data to show that some of this work is really good. Anybody else? I only know of a few labs in the country that do actually a couple labs. I believe there's one lab in Austin, Dr. Cavusi's lab in Austin. I think it's Cavusi. They do DNA fragmentation on everybody. And the only reason why I know that is one of my students is interviewed to go work there. And so we want to teach them how to do some of that work. It's not part of our normal andrology training because not everybody does it. But we do a lot of it in our research part of our IVF lab, in our andrology lab, or our training lab. I also know a lab out in New Mexico that does a lot, in Albuquerque, that does a lot of DNA fragmentation. They might actually test every single patient. The real challenge with doing these tests in your lab is that if you're going to do like a halo assay, or if you have the equipment to do the tunnel assay, I don't think anybody, I think what Don developed with the sperm chromatin structure, the flow cytometry, was his proprietary type system, but most of us don't have that in the lab. So what could we do in the lab for our internal DNA testing? And that's what we do with the halo assay. The tests are a little bit expensive. A kit costs around $800, and there are 10 tests in the kit. So it's going to cost you $80 per patient. I actually worked with a lab, the Give Legacy lab, where we were doing the DNA fragmentation and using their computerized assisted semen analysis system to read them. Until I had my first CLIA inspection, come to find out, the CLIA inspectors know that these tests are not FDA approved, And unless you say you're doing it for research, they want to know what it is you're doing for research. If you're reporting a result to a patient, you need to do your own internal assays, proving one, its efficacy, and proving that your tests are accurate and precise, very much like how anybody would have to do for, getting it FDA approved. I would say that most CAP inspectors probably don't realize what that is. They just want to see, are you running a control? And are you running controls on that? Are you running some sort of proficiency testing? Much like semen analysis, it's not technically a regulated assay, but you would have to develop, if it's an internal derived test, you would have to develop your own internal systems and show the compliance people, rather be CAP, Joint Commission, CLIA, whoever's doing your inspection of your andrology lab, that you are doing these tests, that you have your internal controls. But something that I did early on when it first came out was the HbA slide. And I always wondered if the HbA slide, I believe Cooper sells the HbA slide. I have no commercial interest in any of these companies, but I used to do the HbA slide just to kind of do an internal, just quick test. So I didn't charge any patients for it. And these slides are fairly expensive. I want to say they're anywhere between $50 and $100 a piece. I'm not exactly sure. So, but you can actually just put sperm on there, cover it with a cover slip, and some sperm will stick to the slide and some sperm will be moving around. And interestingly, we actually thought, well, let's put some sperm on there after the Zymot prep one day. And we put the sperm on there. In fact, we used the Zymot and the CA0 derived sperm, the same sample. The pre was, it had moving and stuck sperm and not stuck sperm. So let's say it was 20% DNA frag. I don't remember exactly, but post prep, 100% of the sperm stuck down that slide. Like maybe only one or two sperm were moving around, and both preps. And so it tells me that HbA slide, it's a hyaluronan binding assay is what it's called. Is that slide actually probably would be a really quick internal test. Now, I'm not going to encourage anybody to do this and try to charge for it. If you're going to do this test, it's something that you could just do as an internal, kind of like testing the pH of your media. And, is there something there? Maybe you need to dig further into it and maybe send it off for an actual assay. It might be a way of doing just a real quick internal test. Anybody here ever done the HbA hyaluronan binding assay test in their labs? No. So yeah, I guess if you were to decide to do some of those, you'd have to make sure you're validating those studies. All right.
I had a quick question for Scott, actually.
Yeah. Yes,
Scott,
please.
you mentioned that you give your patients an option to do the Zymot. Do you guys show them data and kind of educate them?
Well, they're counseled by the physician. Just looking over this data, we have two patients that have kind of piqued our interest about this SCSA. And my argument is, let's just use the Zymot, because if we can see that it almost creates a 0% or negligible percent of DNA frag, then it would make more sense just to use the Zymot. So I guess the longer answer to your question is, we don't personally here in the lab, the physicians do, and I think it ends up being a cost thing. But for our cost, what we add, what they add for the Zymot is insignificant in my mind. I think that just using the Zymot versus the getting the SCSA is a more productive way to do it. They have to sign a consent and to use this device. So that's why I think we have such a low percentage of patients that end up using it. I kind of wish we had more, especially if they're doing ICSI. We are not 100% ICSI center. We are probably, I would say almost 50-50. I've always been in the camp that if things can fertilize normally, let's go for it. But this data is kind of hard to argue against sometimes. So I'm happy that, Dr. Anderson presented this data.
Thank you, Scott.
Yeah, thanks for the question, Doug, and thank you for the comments. I've actually been thinking about doing less ICSI and more conventional. And part of the reason why I do 100% ICSI in my labs, because I feel like I'm maximizing fertilization, and I'm minimizing triploidy. But I would tell you, I mean, just as a word of caution, if you're going to use one of these devices, I think you'll still get some of the same benefit, or if you did conventional INSEM with these guys, but you do have to wash the sperm after a swim up. You have to wash the sperm after a Zymot, because there is some residual seminal fluid there, and that is going to prevent the capacitation of the sperm, and it won't continue on to acrosome reaction. So you'll get no fertilization if you don't wash the sperm. So, I think it's important to note is, like, we said, where does DNA fragmentation come from? And it comes from, we call it reactive oxygen species. And everybody here has heard that, right? But what does that mean? It makes us sound really smart when we say it, like, well, it must be the reactive oxygen species that cause the DNA fragmentation. But what does that mean? And really, if you think about it, it just really means an extra oxygen. And so when we get the sperm in the ejaculate, we have it in a large cup and a large base bottom that is exposed to more air, and that's kind of the science between the Protex cup is Sam Preen has developed a cup. It's an expensive cup, but the idea is that the sperm is in a very low conical space, and that sperm is now in a very low environment. And it's actually, I call it the Yeti for sperm, because it's got a little foam base around the bottom of it. I get donations to the IVF Academy all the time, and I had these specimen collection tubes. They're literally like conical tubes, probably almost like a 10-15 mil conical tube. We all have these in our labs, but it had a funnel you could attach to the top. So the guy would ejaculate into that funnel, very much like ejaculating into a cup, but then it would go down through the funnel into the tube. So that might be another way of actually reducing the exposure to oxygen, because we live in an oxygen-rich environment. The oxygen we're breathing is 20% oxygen, whereas in the body, we culture our embryos at 5% oxygen, because that's closer to our physiological oxygen level. So when we the sperm is ejaculated, it's exposed to this oxygen-rich environment. So we are potentially exposing it to risks of DNA reactive oxygen species. Some of the early papers that a lot of people said, the reason why I'm bringing this up is if you're going to wash your sperm after a Zymot prep or a CA0 prep or Novasort prep, then you're centrifuging it. And a lot of early studies showed centrifugation caused DNA fragmentation. In fact, Aiken was some of his first, his papers back in I believe it's like 1978 or like early 80s, late 70s, showed that DNA fragmentation, higher levels of reactive oxygen species, not so much DNA fragmentation, levels of reactive oxygen species increased after centrifugation. So, but if you go read the paper, they were spinning it multiple times at really high speeds. Like, I would almost like to redo that paper. And this is where, like, I think it might even be a really great, another really great PhD paper. If anybody here is working on their PhD at EVMS or another institution, go back to some of these early papers. try to redo some of that work. I would imagine that, you know, spinning it at 300 Gs, which is only around 1500, 2000 RPM, not like 4000 RPM. You only spin it one time, or how many times do you have to spin it before you do actually cause damage, like some of this work? And just, you know, I would encourage the audience to push forward on some of those things. But I did some, I was doing some work with a group that was freezing their sperm, and we were shipping it out overnight. So we would, the guy would collect, we would use our internal semen analysis. We would do a DNA frag when they collected, ship it overnight, centrifuge it down, and then freeze it. There really was not so much increase in DNA fragmentation from the shipping, because the seminal fluid actually is protective of that sperm, you know, while it's sitting in it. We would dilute it in some media so we would have good survival. But so we'd have it survive overnight. We really didn't get really increases in DNA fragmentation through the centrifugation. But where we did see significant increases in DNA fragmentation was the frozen sperm. So one of the things we do, if we're freezing sperm for backup, I guarantee you it might have been, you know, 20% DNA fragmentation pre-freeze, but post-freeze, you're probably getting up there into that, you know, 30% DNA fragmentation points. And so kind of consider that, especially if you're using some of these devices like the Zymot or the NovaSort. If you're not getting great recovery, it's probably because the DNA fragmentation is high. So you know, I get people call me when if I was talking about the Zymot device or something, like I get really terrible recovery for my donor sperm. Well, that's okay, because you only, if you're doing ICSI, you only need 10 sperm for the 10 eggs you're working with. So, but you're going to know all 10 of those sperm are good. So that's just kind of, you know, I think, you know, as someone mentioned, I wish we did more, because you're right. You're not going to have to do that testing with it. You know, SCSA or any other outside tests. So any other comments? I'd love to hear from anybody in the audience. Please feel free to share. So we're kind of getting to the end of the I don't want anybody to go over time. I'm going to let Lygia talk a little bit here. One of the things I would like to do is please share some of your ideas that you would like to, some papers you like to read. I believe the next time somebody shared a paper here a couple weeks ago with me talking about microplastics in the follicular fluid. So that's going to be probably our next paper. And we'll look forward to that. I hope you'll come back and join us again. We're going to do this every other week. And thank you for joining today. Take it away, Lygia.
Thank you. So we also do have some short intensive classes coming up covering ICSI, biopsy, vitrification. Those are 2 1-day or 2-day courses. And then we've got a week of embryology and a week of andrology coming up. As a participant today, I can offer you 25% off those classes. So I will be emailing you with that information, and I look forward to connecting with you all and seeing you all at the next Journal Club. Thank you.
Thanks, everybody. See you soon.